So every cell in the human body has essentially the same genetic information, our DNA, which encodes the information for how life works. But every cell also does things differently. For example, my eye cells are very different from my skin cells, are very different from my stomach. And how that information that's encoded on our DNA strands is translated into how the cells actually function is performed by a class of proteins, little molecular machines called transcription factors. These transcription factors bind to the DNA and read out the information and tell our cells what to do. And stem cells can turn into any kind of cell, and the reason they can do that is because these transcription factors are doing different things. Similarly, cancer is usually caused by a mutation, a very small change, which causes these little machines to act differently. When they act differently, it changes how they work, and instead of being a skin cell, it starts going crazy and you get melanoma. So this understanding of how these little molecular machines, these transcription factors, act to bind DNA, to recognize it on specific portions and do their jobs, a better understanding of that and a better way to track how that's happening in real time will let us understand these important tissues much better.
Okay, there's two challenges related to the sensor design in general. We can compare the sensor design challenge by going out fishing. At some point, if one wants to detect different transcription factor, in the image, transcription factor can be fishes, one needs a good bait. So, and the second thing is that if you throw the bait in the ocean, you won't be able to catch any fish. You won't be able to monitor any fish. So you need to come up with good baits as a good biosensor, and you need to come up with a signaling mechanism. You need to come up with a strategy where you're going to basically see the transcription factor or the fish binds to the bait. So this whole project was inspired by nature because we soon realized that all the baits necessary to detect transcription factor are already encrypted in the DNA of any cells. So these transcription factors, so let's say this is a DNA and this is a transcription factor, we already know that transcription factor binds specific DNA sequences. So by knowing these DNA sequences, we now have the baits to detect those transcription factor. But now the second problem is that we can synthesize those DNA strands, but when the transcription factor will bind those, there won't be any signal, right? So it's like putting the bait in the ocean, the fish will eat the bait, and we won't know if the transcription factor is present. So the second thing that we were inspired by is by nature nanosensor. Actually, our body is made of contains billions nanosensor. These are small machines that are able to detect molecules. For example, in our nose, we have like millions of these switch molecule, which when they bind odorant molecule, they change their shape. And this change in shape is the information necessary to say to the brain, oh, the odorant molecule is there or not. So we use the same exact strategy as our signaling mechanism. So we took those single-strand DNA, we took the DNA sequence that are known to bind transcription factors, so we took these baits, and we just re-engineered them so that they become molecular switches. So, for example, if this is like a DNA sequence that is known to bind a transcription factor, we just redesign these DNA so they adopt another structure. With this structure, the transcription factor cannot bind, but when the transcription factor bind, the structure open up. And the next step, we just engineer optical reporter on that DNA strand. So when the DNA is in that conformation, there's no light. But when the transcription factor binds the DNA, there's light. The optical reporter are very sensitive to the structure change, and there's light emitted. So, in summary, if we want to detect transcription factor with our approach, we just take cells, mash them up, put our sensor. If we see light, that means that the transcription factor is present. If we don't see light, that means that the transcription factor is not present.
Stem cells are cells whose fate is still open. A stem cell can develop and turn into a muscle cell or a neuron or a cell in your eye. Cancer cells are cancers whose fate was once determined, but then gets broken and they start to grow and proliferate beyond what their normal stop point should be. in both cases, the fates of these cells are defined by the concentrations of these transcription factors, by the concentrations of the proteins that we are detecting. These proteins are in effect the control points, the set points for the cell. They're analogous to the knobs on a console. And by measuring the concentrations of these, we can tell what the knob's set to. Is the knob set to 5, which means the cell is going to become a nerve cell? Or is the knob set to 9, which means it's going to become a muscle cell? Or is it set to 11, which means it's going to become some cell in the eyeball? Being able to measure the concentrations of these transcription factors is analogous to measuring where the knobs on a console are set. So it'll help scientists understand better how these control points control the programming and fates of cells, either stem cells to develop into new tissues or normal cells to develop into cancer.
The answer to the first question is absolutely. The transcription factors, which are the target of this sensor technology, are very much involved in cancer. So any approach that can interfere with that is very likely to have a consequence on cancer development. So absolutely, and there's actually precedent for these. There are a number of drugs that are out there already that interfere with those proteins. This technology can just make that process much faster. Now the second question, the second part of the question has to do with how long will it take? Well, drug development is a slow process. In general, it takes around 10 years to get a drug to market. But this technology is focused on the front end of that process, on the basic research side of things. And really what it has the power to do is to make that process faster and make it less likely that what is discovered will have side effects. And that's really an important contribution of this technology.